be introduced to is the idea of symbiosis and evolution. They're both closely related, and a lot of times these symbiotic pathways can have huge implications on the evolution. Now, symbiosis in nature is when you have two organisms or two species acting really closely in a close relationship. So we see this terrestrially with plant pollinators, but we see this a ton of the time in the aquatic world. Whether you see this in your anemones and your clownfish, or we have these cleaners that will clean off ectoparasites on plant species. But where this is really interesting is with these goby-type fishes that perform a lot of different symbiotic interactions. As you can see from these images, they are very diverse in their shapes, their colors, and all of their body morphologies. But who is the family Gobiidae? Now, it is a massive group of fish with more than 2,000 species and more than 200 genera. And while they're really massive in the amount of species they have, they're actually very small-bodied fish that live in benthic areas. And within this high diversity and the high amount of species that we see in this family, it opens up a bunch of different diverse niches and microhabitats. Arguably one of the most interesting microhabitats that they employ is actually in the burrow of a shrimp. So the shrimp-goby mutualism is a relationship in which the shrimp will construct a burrow for the goby and the shrimp to live in, and in exchange for having a nice home, the goby will watch out for predators at the entrance of the burrow and will actually alert the shrimp if there's predators, because the shrimp tends to be completely or at least a little bit blind. So the huge crux of this relationship is the fact that they are living together in this burrow. While I said gobies are pretty small-bodied fish, these burrows are not very large, and so you'd have to imagine that maybe there's some size selectivity on how big the goby fish wants to be to maximize this relationship that they're living in this burrow with the shrimp. So, with the idea of size selectivity in mind, I present to you this continuum of body elongation. So up here are two fish that were in my sample. We see this really, really tiny one with maximum 16 millimeters, the thinness. But we also see this huge one at 350 millimeters that are occupying two ends of this length spectrum. But what I want to know is, where are the shrimp gobies? Are they really small like this to fit in the burrow? Do they maybe need to be a little bit bigger? Where are they on the spectrum? While body length is a pretty easy continuum to wrap your head around, body shape gets a little bit more confusing. We have this fish over here, Gobiodon, which is very short and it's laterally compressed. And that's often because they live in association with branching corals, so that makes this body shape really advantageous. But we also see the other side of the spectrum in the genus Microdesmus. They're extremely long, but they are straight up shaped like a tube. They're extremely fusiform fish. So again, I want to know, where are the shrimp gobies in their body shape, and does it have anything to do with the fact that they are living in a burrow? So going back to symbiosis, there's two main things that can happen. The first, as we see from these pictures here, is a huge array of diversity. You can be any body shape, any color, you can have different morphology, you can have these really interesting different types of habitats like in these corals. So with more radiations and new diverse adaptations, we get huge diversity with these fish. On the flip side though, you could be a really diverse goby and become symbiotic, and over time you slowly start to get constrained until you're at this optimal body shape. And with this, you wouldn't be able to become really diverse because you've wholly optimized yourself to be in this symbiosis. So here I propose the question to you: does being a shrimp goby constrain, or might it actually promote the evolution of shape change? So from here we have this hypothesis. Well, if evolution is constraining shrimp goby shape, we expect that shape disparity will be smaller in shrimp gobies. So this is visually represented here. You can think of all of these dots as different species. All of the shrimp gobies, their body shape is going to be really constrained or really similar. They're not going to have wide variation in what they might look like. But if you pick any species of a non-shrimp goby, they can be really small, they can be really long, they can have a lot of diversity. Now this is what we expect to see. So if evolution is constraining the shrimp goby body shape, we would expect that shrimp gobies have their optimal body shape that works really well in this symbiosis, versus the non-shrimp gobies that are going to have a really distinctly different body optimum that they are evolving towards. So with this idea of constraint, we also have to think about why are they constrained? And that gets to the idea of selection. So we can model this in two different types of models where we expect to see two slightly different things. Our hypothesis is that there is in fact two distinct optimas, which would mean shrimp gobies are constrained and have to have one type of body shape. The alternative, though, is we could see this single optima selective model in which there's one distinct body shape. No matter what kind of goby you are, shrimp goby, non-shrimp goby, selection is actually going to pull you to one body shape for everybody. So we also need to think, well, if evolution is not constraining the shrimp goby body shape, what might that look like? So we tested a Brownian motion model, which basically states that there is no selection going on here. So there's no distinct body optima. You could be a shrimp goby, you could be a non-shrimp goby, you could be symbiotic, you could be anything in between, and you can primarily do whatever you want with your body shape. You're not constrained by selection. So with this, to understand what's going on here, we asked three main questions. First, is shape and disparity different between shrimp and non-shrimp gobies? And the method I used here was geometric morphometric shape analysis. Next, we need to know, well, where did this mutualism come from and how did it evolve in the phylogeny? So we built a phylogenetic tree and employed ancestral state mapping. And lastly, we need to know, is shape change in shrimp gobies evolutionarily constrained or not, and is selection present? So to do this, we fit varying types of models of trait evolution. So the first thing that we did was this photo collection and analysis. I had 86 gobies in the family Gobiidae in my sample set, and I went online and tried to find images that looked kind of like this one, where I could see all the morphology, it was a clean shot, and it also needed to have the standard length, because I needed to know how big this fish was. If we couldn't find that, we ended up employing the Smithsonian, who sent me a big box of fish that I was able to photograph in the UW fish collection. Once I had all of these images, we then went and landmarked 17 different points on the computer, so all of the fish had these little red dots on them. Now these represent just the general body plan and morphology of all gobies. So once we were able to quantify what is the general goby body shape, we plot it onto this. So this is a principal component analysis. And what this graph here is showing you is in red, the shrimp gobies are all pretty clustered on that middle point. And we see the blue, non-shrimp gobies, have a little bit more variation. And each of these squares represents one of the 86 species in my sample set. So our first question here is, are shrimp gobies and non-shrimp gobies different in body shape, yes or no? And we found this to be statistically significant, that they are in fact a different body shape. One important thing to look at on this PC graph is the horizontal axis, or PC1. So this here we found is actually measuring body elongation. And this 50% is saying that that variation in elongation is 50%. So a pretty significant portion because of the elongation. We see this visually here again with Gobiodon, which is really short and stubby and laterally compressed, occupying this square here. And we also can compare this to the other end of the spectrum, Microdesmus, which species are represented by these circles. Super, super long and tube-like in shape. So we see again that this is statistically significant that non-shrimp gobies have more diversity or more disparity, which supports our hypothesis that there is a constraint on the shrimp gobies. They have less morphological disparity. So from here, this is where we show where the phylogeny has the mutualism showing up. So each of these tips are different species in my sample set. We see three main groups have this relationship with shrimp. We see it in the silt shrimp gobies, the reef shrimp gobies, and then interesting, we see Ness longus, which is a part of the American seven-spine gobies, and is the only goby in that group to participate in this relationship with shrimp. Now here's the results of our modeling. So we see this delta AIC score. You can kind of think of this as a best fit score. You want this score to be really low. So we had really low low values for the delta AIC for our selective models. And this looks like that 2 optimum model or single optimum model. The one that fit the best supports our hypothesis that yes, there are 2 distinct body shapes that selection is pulling gobies towards. You have your shrimp gobies occupying one body space, and then you have your non-shrimp gobies occupying the other. However, we did see that this single optimal body shape also fit equally well. Scores below 2 are usually really supportive, and both of these were below 2. So that means that both of these models are showing us that selection is present. We can't really rule out one or the other. What we did find is that it's definitely not a Brownian motion, because these scores were absolutely huge. So this really implies that Gobi shape is under selection. They can't do whatever they want. They can't get really long. They can't get really short. They have an optimal body shape somewhere in there. So to explain further this 2 optima model, it really means that your shrimp gobies have one shrimp goby shape, and this so happens to be really advantageous in this shrimp goby mutualism. And on the other end of the spectrum, you have these really diverse goby forms and all other members of the family Gobiidae. It's 2 distinct body plans. But because we saw equal support for this single optima, that means all of your really diverse gobies, your regular gobies, your shrimp gobies, they're all evolving towards one optimized, perfect goby body shape. It just so happens that these shrimp gobies, this really works for this relationship, that whatever all goby optima is, shrimp gobies can do that too and live in this burrow. Now, what does this all mean when we put it together? So we found that selection is a really huge driving force on shrimp gobies because of what we found in our modeling. Those Brownian motion models fit extremely poor, so we know selection is definitely happening. We also know that elongation is a huge factor in this relationship based on what we saw from our morphological analysis or our PCA. However, it's a little bit unclear about who's actually constrained here. If we think it's the 2 optima model, that supports our hypothesis that shrimp gobies are very different than non-shrimp gobies. But if we think about this single optima model, maybe shrimp gobies are constrained, maybe it's actually all of the family Gobiidae. These fish can get really, really small. There's this idea that miniaturization might actually be pushing this constraint. It's hard to be a small fish, so maybe that in itself is what the constraint is, and it's not so much the fact that they're symbiotic. Lastly, I propose that maybe it's actually kind of both models. Maybe we see that it is a single optima where everyone's evolving towards one optimal body shape, but maybe these shrimp gobies just have slightly more likelihood to deviate because of the needs for their bodies to fit inside of these burrows. So for future work, one thing we could do is try to increase our sample size to see if maybe that pushes that significance of that AIC score and selects one model over the other. However, we had a pretty robust sample size with shrimp and non-shrimp gobies. We could also test for coevolution because this showed up multiple times in the phylogeny. That might be a good way to understand how the evolution of this trait is acting on morphology as well. And then one thing I think would be really cool is looking at other symbiotic groups, because gobies employ a lot of different types of symbioses, especially with corals. So we could do the same methods with obligate coral-dwelling gobies that pop up multiple times in the phylogeny, and kind of compare and contrast that work with our study to see if symbiosis is constraining in other types of symbiotic relationships within the family Gobiidae. So with that, I'd like to thank these people here. Thank you, Luke, for helping me with this project. I'd like to give a huge shout out to Liz Miller, who is a postdoc at UC Irvine and also went to UW for a short time. She helped me greatly learn how to use R. I'd also like to thank my friends at the UW Fish Collection, Catherine and Kat, for teaching me how to handle my Smithsonian loan and just learn all of the ins and outs of fish collection. And then also thank you to the Smithsonian for sending me a very big box of fish. All right, anyone have any questions? Dr. Husband.
No, you go with
Okay.
So with the PC plots, going back to those, so the x-axis was length, and it seemed like the ones that were longer were the non-shrimp gobies. Was that, am I getting that right? And if I am, is there a reason that the really long ones are the non-shrimp ones?
Yeah, they definitely had more of a tail. There were more of those. So I had 3 of the Mycodesmus in my sample, so it could be that they're just maybe pulling that over that way. But in general, there's just such a huge diversity in gobies. You do see a lot of them that have this kind of strange worm-like body shape. Yeah, it is pulling it a little bit in that way where there's more on the elongation, but I think that might just be due to the sample size that I had 3 of them.
It's really cool stuff. Question, does every goby species have a symbiosis with one specific shrimp species, or do they just go in with any shrimp?
So from what I understand, so there's many different genus that will pair with shrimp, but it tends to be only, I believe I'm saying this right, Alpheus shrimp are the ones that will do that. And so not every, I think it's only that one genus of shrimp that's gonna pair up with gobies, but there's like maybe about 10 different genus, I think, that will do this with the shrimp.
So is there correlation between the shrimp body shape and the goby body shape?
That's one thing I think would be interesting, and I had a hard time finding papers that talked about the shrimp in association, because I think there's really something going on with the whole operation of the burrow. I did find that gobies and shrimp are pretty selective in who they pair with, and so there was a study that put a bunch of different known shrimp gobies with one type of shrimp. Not all of those shrimp will pair with all of the gobies. They kind of have their preferred genus. So I think more work should be done in understanding the shrimp component. Right now they seem fairly separate.
Cool.
Yes.
How do you tell if it's a shrimp or a no shrimp goby?
So there's different genus that definitely perform that, and so you could have, like the genus Amblyotris, all species in there are gonna perform it. So if you can ID it down to the genus, you know that that'll be a shrimp goby.
Oh.
Yes.
Thank you.